So now let us quickly dissect the flower and see the structures present inside it. So for you all, I have plucked one flower, but you should not pluck flowers unless it is absolutely necessary for you. And apart from the flower, we have some clean and dry slides here. These are just rectangular pieces of glass. If you don't have them, it's absolutely fine. And I have placed the slides on these filter papers. If you don't have filter papers, you can use simple plain papers as well. And then we have a watch glass, and in the watch glass I have taken some water. Then we have a forceps with a pointed tip, and I am going to use another forceps also, but one is absolutely sufficient. So now let us just start. So let us observe our flower carefully. Now this stalk which joins the flower to the stem or to the branch of the plant is known as the pedicel. This stalk-like structure is known as the pedicel of the flower. With the help of this pedicel, the flower remains attached to the branch or to the stem of the plant. And you can see that here are small birds as well, flower birds. Okay, so above the pedicel, we have this green sheath-like structure which is known as the calyx. And each unit of the calyx is known as the sepals. These pointed tips, these are actually the sepals. Can you see here that each of these sepals are actually joined in case of this plant? This plant is hibiscus. And in hibiscus, we find that the sepals, these pointed things, these are all joined together, isn't it? We have a one single structure here. So these sepals are all joined together in hibiscus. Another interesting thing that we must note here, that in flower birds, this is a flower bird, okay, and in flower birds the calyx covers the entire bird. It houses the entire bird. Why? Because the calyx provides protection to the developing bird. Calyx has a protective function. It is protecting the developing bird, and that is why it is covering the whole of it. Okay, and what is this thing below the calyx? These things, pointed things, these are called epicalyx. Here also in the flower also we can see that the epicalyx are present. Okay, so we learned about calyx, and then we learned about epicalyx. And what is the function of calyx? The function of calyx is to protect the developing bird. Now let us count the number of sepals that are present here. So we have one sepal, then two sepal, this is the third one, this is the fourth one, and this is the fifth one. So we have 5 sepals in case of hibiscus. Now let us quickly count the number of petals. What are petals? These colorful, vibrant structures are actually the petals. And just like sepals, we have again 5 petals here. 1, 2, 3, 4, and 5. Now the question is, what is this structure known as? So let us see. At the top we see that there are 5 branched tips. And at the top of each branch there is one globular red dot-like structure. These globular red dots are called the stigmas. Now in hibiscuses we have 5 stigmas, 1, 2, 3, 4 and 5. And this stalk which is holding the stigma is known as the style. So here we have a branched style, and we have 5 branches of styles, 1, 2, 3, 4 and 5 again. Okay. Now what does the style do? Actually the style runs all the way down here and it reaches below here, because here we have another part which is known as the ovary. So the style runs all the way down and reaches the ovary. The style connects the stigmas to the ovary down below. We will see the structure of ovary in a little while dissecting the flower. Now what are these called? These yellow dot-like structures are called the anthers, and the stalk which are holding the anthers are called the filaments. And this entire thing, the anther plus the filament, they constitute the stamen of the flower. Okay, so far we have studied 4 structures. 1, the outermost whorl of the flower, the outermost structure of the flower, which are the calyx, and each unit of calyx is known as sepals. Then we have our corolla. This structure, this is known as the corolla. This is the second wall of the flower. This is the corolla, and each unit of corolla is known as the petals. Then we saw our stamens. These yellow dots on the stamens are actually the anthers, and the stalk holding the anthers are the filament. And then we saw our innermost structure, which are the pistil. Now in the pistil we have this stigma and then the style, and below we have our ovary somewhere here. Now it is time to dissect the flower, and for which I am going to use a blade here. You can use any blade, but be careful while handling the blade. So first we will cut open this calyx, which is the outermost wall of the flower. Okay, so I have cut the calyx, and I will try to separate the be very gentle, otherwise you might break the entire flower. I am trying to gently remove this calyx from the flower. Yes, it is here. We have our calyx just here. Now I am going to place the calyx in our water for some time. Remember the watch glass that we had taken? Yes. So I'm going to place it there. Now here we see that we have the petals completely exposed, and these petals are not joined with each other, are they? No, they are all free here. See, they are all free here. So the petals are not joined in hibiscus, but the sepals were. The sepals were joined, but the petals aren't. Okay. Now I have not removed the epicalyx yet. We will do that later. First we will try to remove or separate the petals one by one. We can simply do that with our hands. Just hold at the base and gently pull out one by one the petals, just like that. And the fourth one is removed, and then the fifth one as well. So here finally we have our 5 petals. And after removing the petals, we have a structure like this. Now some of you might think that this is the style of the plant or the flower, but no, this is not the style. In hibiscus, this is not the style. In other cases, it could be, but in hibiscus, it is not. Why? Because in hibiscus, we have a condition which is known as the epipetalous condition, and this is a monadelphous stamen. What does that mean? Monadelphous stamen means that the filaments of our stamens are all joined together to form this sheath, this tube-like structure. So now our job is to cut open this tube to expose the female reproductive part or the pistil. For that, I will take a slide here. Be gentle, do not make a very deep cut, otherwise you may end up cutting the ovary only, and we do not want to cut the ovary. Now try to separate it with the help of a needle or a forceps, whatever you are having. I am doing this with a forceps. We will split open the entire tube, but please be very gentle while doing this. Okay. Very, very gently. You can use the finest needle that you have. Now let us try to open this ovary. Yes, this is our ovary is exposed. Now we will very, very carefully remove our stigma from this monadelphous staminal column. Sorry, style. We will remove our style from this monadelphous staminal column. You can see that the style is coming out. You can see how thin the style is, and it can break anytime if you are not extremely careful. Yes. Be careful that always try to keep this in water, otherwise it will dry up really quickly. Now that we have completely taken out our pistil or the female reproductive part, so let us clean this ovary a little bit. So this part, this swollen part is the ovary, and I am going to remove the epicalyx and the other parts. But before that, I want to mention a very interesting thing here, that we can see that the epicalyx are arising from below the ovary, isn't it? This is our ovary and the epicalyx was below it. Or let me put it in a different way. I think now it is easier for you to understand that this is the ovary and the epicalyx, these parts are the epicalyx, okay, and the sepals were also arising from here only. So we have our ovary above our sepals, petals and the epicalyx. That is why this is an example of superior ovary, and this condition is known as hypogynous condition or hypogynous ovary, superior ovary or hypogynous ovary. Now I will just cut and remove this epicalyx portion. Okay, I think I have removed a little bit. Yeah, it is done, and I will place it in water. And here we have our complete female reproductive structure or our pistil. At the bottom we have our ovary, and this ovary is then connected to the style. This thin fine filament like structure, this is the style. And above at the top, it is the style is branched to form 5 structures, and these 5 structures are connected to the stigmas. These dots at the top, these are the stigma. This is the place from where the flower receives the pollen grain. Okay. After that, we will talk about our male reproductive part or this monadelphous stamen that we have found in our hibiscus plant, where we have our staminal column. This is the tube-like structure. This tube-like structure is the monadelphous staminal column, where the filaments have joined together to form this tube, which covers the style of our pistil. And then we have our anthers here, these yellow dot-like structures. We have the anthers here. Now, if you are going to sit for a practical exam and you need to preserve your pistil intact, but also you need to produce a TS, a transverse section or a longitudinal section of your ovary, then you can simply use a flower bud. Just hold it like this, and at the bottom of your flower bud, just put a cut mark. Very fine cut, as fine as possible. Now this fine cut is actually the TS of your ovary, transverse section. If you want a longitudinal section, then you can cut it in a longitudinal way. I want a transverse section, so I have cut it like this. Now this is the TS of your ovary, which you can obtain simply from a flower bud by cutting at the bottom, at the base of the flower bud. Okay. Now that we are done with our dissection, it is time to arrange all of this on a sheet of paper and label them properly. This labeling and arrangement is very important. So first I have placed our calyx. After that I have our corolla, and I have also mentioned the number and their arrangement, which is in this case it is joined. Then we have our petals or the corolla. Again I have mentioned the details like it is free, they are all free. And also another important detail about the corolla of hibiscus, that the petals in hibiscus, they remain joined to the staminal column, and this condition is known as epipetalous condition. Okay, then after that we have this monodelphous staminal column or the androecium of the flower. Then we have separated some anthers and I have put them on a slide in some water, and we have presented that too. Then we have our TS of ovary, and then we come to the pistil or the female gynoecium, and again I have labeled it stigma, style and ovary. So that's all. So that is all about flowers. I hope you have learned something from this video. Thank you.